first of all, I'd like to start by thanking Sapna and Brent for organizing a really wonderful symposium and for the opportunity to share some of our work on wintering birds. And I want to talk today about some patterns and mechanisms of seasonal and among winter and within winter metabolic variation in small birds. And we focus on birds for one reason, because they're endotherms and they remain active all winter. So we've heard several talks over the last few days about organisms that go dormant or kind of downregulate metabolism in winter. And that's a very different response than what we see in birds that are active, that actually have very high thermogenic costs or thermoregulatory costs in winter. And so they upregulate metabolism to deal with that. And we focus on small birds, less than about 50 grams of body mass, because they have relatively high surface area to volume ratios. And as a consequence of that, they have high rates of heat loss and have to respond to that with greater flexibility in metabolic rates. And so I'll start with a definition, and that's of what we mean by phenotypic flexibility. And this is a subcategory of phenotypic plasticity that deals with temporary reversible changes in the phenotypes that usually occur in adult organisms. And so birds, for example, can toggle back and forth between a summer phenotype and a winter phenotype as conditions dictate. And this, of course, allows better matching of the phenotypes to prevailing environmental conditions. And this matching can have fitness consequences. And so the two panels below are two separate studies on black-capped chickadees that look at the relationship between summit metabolic rate, which is defined as the maximum heat production capacity in the cold for birds, and overwinter survival. And those birds that had high summit metabolic rates, at least above threshold levels, were those that also had higher overwinter survival. So it does have some fitness consequences. Here's an example of seasonal flexibility and summit metabolism that we derive for American goldfinches. And the bars there represent the summit metabolic rates in the different seasons in terms of the oxygen consumption. And so the winter birds, you can see, have about 35% higher summit metabolic rate than the summer birds do. And that's correlated with an improvement in cold tolerance as well. So the right-hand axis there is the temperature at the cold limit, which is defined as the temperature in the helium-oxygen gas mixtures that we use to generate summit metabolism that cause the birds to go hypothermic. And so you see it takes a lot colder temperatures in the winter to get birds to go hypothermic than it does in the summer. And that improved cold tolerance is thought to result in that increase in overwinter survival in birds that have high summit metabolic rates. So that's an example of variation between seasons. But metabolic rates can also be flexible among and within seasons. So these are some data that we generated several years ago for black-capped chickadees, dark-eyed juncos, and American tree sparrows. And so let's focus on the left-hand panels to begin with. And the bottom panel is the mean minimum temperature over the period where we measured metabolic rates in these birds. And you can see that in the winter of 91, 92, it was relatively warm. The winter of 94, it was cold. And then the other winters were sort of intermediate. And if you look at the basal metabolic rate and summit metabolic rates, essentially the patterns are mirror images of what you see in the minimum temperature. So birds in cold winters tended to have high metabolic rates. Birds in warm winters tended to have low metabolic rates. And then when we further looked at this within the winter, so the right-hand panel there looks at temperature variables prior to the date where we measured metabolic rates. And so short-term variables were temperature variables within 7 days before we measured metabolic rates. The medium-term temperature variables were 14 to 30 days before metabolic measurements, and then the long-term measurements were 30-year average temperatures. And for all three species, for both metabolic rates, the short and the medium-term temperature variables explained more of the variation in these multiple regression models than the long-term temperature variable. So birds are responding to fairly short-term temperature variation. And here's a couple more examples of that. The left-hand panels are from black-capped chickadees in a study by Magali Petit and Francois Vizina. that looks at the changes in basal and summit metabolic rates over the winter. And you can see they tend to increase throughout the winter as the temperatures get colder. And in the bottom panel that looks at summit metabolism, in February of 2010, it was a colder winter, a colder February than it was in the 2011, and you saw higher metabolic rates there. So they're responding to among winter variation in temperature there. And then the right-hand panels are some data on dark-eyed juncos from Maria Steger with an acclimation experiment. And what I want to point out here is that on the bottom panel that looks at summit metabolic rate, they elevated summit metabolic rate by approximately 20% within a week, and then it remained stable for the rest of the 9-week acclimation period. So they're responding again fairly rapidly in terms of organismal metabolic rates, and that's consistent with what we found earlier in the response to short and medium-term temperature variables. So it's quite a flexible response. And so our lab is interested in looking at some of the mechanisms underlying these organismal responses. And we take an integrative approach to do that. And today I'm going to talk mostly about organ level changes and then cellular and molecular level changes. And when we do that, to focus on birds, the name of the game really is heat production. And most of that in adult birds is thought to be by shivering. And so it's a function of skeletal muscle metabolism. And so we need to look at the skeletal muscle to see what sorts of changes are going on there that could result in these organismal changes in metabolic rates. And there are really two non-mutually exclusive potential mechanisms for that. One is you can enhance the cellular aerobic capacity, or the ability of the individual muscle fibers to produce ATP by aerobic pathways. And this might be coupled to changes in the substrate or oxygen delivery pathways. Or you could get larger muscles. And so we'll start with the second possibility first here. And these are some data that Francois Vezina and I collated in a review paper in 2015 and showed the data up to that date. And what the graph shows are winter increases in summit metabolic rate in the magenta color, and then in the blue is the winter increases in pectoralis mass. And what you can see here is that in a number of species, they're pretty tightly matched. And in all species, you saw winter increases in both summit metabolic rate and pectoralis mass. And so a common element of the winter phenotype is that they have larger flight muscles. Now, there's some more recent data that suggests that maybe the picture isn't quite that tidy, but still the upshot of this is that having larger pectoralis muscle masses benefit your heat production capacity in terms of your summit metabolic rate. And so the next question we ask is, how is this muscle mass change regulated? And one of the primary candidates for that is myostatin, which is an autocrine paracrine muscle growth inhibitor. So it's produced in the muscle and acts on the muscle. And a little bit about, so our hypothesis was that you should see a decrease in myostatin in the winter. So if you decrease the inhibitor, it frees up the muscle to grow. But in order to test that, you need to know a little bit about the pathway for myostatin action. And so it's produced as an inactive form bound to a propeptide. And there's a couple of cleavages that have to take place. And the second cleavage that activates it is accomplished by metalloproteinases. Prominent among them are the tolloid-like proteases 1 and 2. And those cleave the propeptide from the mature form, and the mature form can then bind to the receptor and initiate the downstream actions that result in inhibition of muscle growth. And so there's two primary points of regulation for myostatin. One is the actual expression or synthesis of myostatin, and then the second is the processing capacity, or the ability to cleave it to activate it. And so we looked at this in a number of different birds and a number of different sort of situations. So the top three are American goldfinch and black-capped chickadee and house sparrow, and those were natural seasonal acclimatization. So we compared summer birds and winter birds from natural seasonal environments. And then the bottom two were house sparrows that we did acclimation studies either to an acute cold stress or to exercise. And then we looked at the myostatin gene and protein expression and the gene expression of the tolloid-like proteases that activate it. And in general, the trend is that these changes, either winter or cold, increase the pectoralis mass in these birds. And along with that, you see decreases in the level of myostatin, either in the mRNA or the protein expression. Now, not all of those changes were significant, but the pattern's pretty similar. Increase in pec mass results in a decrease in myostatin. And then for the naturally acclimatized birds, we also saw decreases in the processing capacity. That wasn't true for the experimental treatments, and the tolloid-like proteases do some other things besides just cleave myostatin. So it may be that the experimental protocols generated some of those effects. But the general take-home message is that these results are largely consistent with the role for myostatin in regulating some of that pectoralis muscle mass change that we see on a seasonal basis. So now I want to move into the second possibility, and that is the cellular metabolic intensity changes. And so this is work that I did with Zach Chevron at the University of Montana, looking at a pectoral muscle transcriptome for black-capped chickadees and American goldfinches that were naturally acclimatized to summer and winter. And both of these species showed discrete seasonal phenotypes, but the seasonal separation was greater in chickadees than it was in goldfinches. So you saw more genes and more pathways that showed differential expression in chickadees than you did in goldfinches. But both species upregulated glycerolipid metabolism, and chickadees also upregulated the citric acid cycle and some other fat metabolism pathways as well. And that's important to note because fat is the major fuel for shivering in birds. So this upregulation of fat catabolism capacity is consistent with the use of fat as fuel in these birds. And then we also did metabolomics on the same individual birds, and the pattern was basically the same. Chickadees, both species showed distinct seasonal phenotypes, but the separation was greater for chickadees than it was for goldfinches. And the winter changes that we saw included a reduction in amino acid levels, and that could be due to decreases in dietary protein intake in the winter, but it also might be related to increased protein turnover that's related to muscle remodeling. We also saw reduced levels of the citric acid cycle intermediate, so that suggests faster cycling to increase cellular metabolic capacity. Activation and biosynthesis of fatty acid derivatives, again, consistent with lipid oxidation. And then also, interestingly, we saw an increase in the NAD pathways. And NAD is a regulator of metabolism, and we think that this relates to an increase in cellular metabolic output as well. So there's also some changes that take place at the cellular level that can influence the cellular metabolic capacity. So both bigger muscles and changes in cellular metabolic capacity seem to occur in birds. Now, one of the questions that hasn't been well addressed for birds, and actually for a lot of other organisms as well, is about cost and trade-offs of these flexible phenotypes. Why don't all birds just show as much flexibility as you can? And the thinking is that there might be costs or trade-offs that are related to that. And some of the costs that have been proposed include maintenance costs, which are costs associated with maintaining the capacity for flexibility. And so this is an environment independent cost that you're going to pay all the time to have a flexible system. And it also includes the sensory apparatus necessary for sensing and responding to environmental change. And then production costs, which are costs of that are associated with generating the flexible response. So that's an environmentally dependent response, and it only is generated when the environmental conditions dictate a change in phenotype. So one way that we could potentially test this is to look at minimum maintenance metabolism, or basal metabolic rate. And so we did that. We collected literature values from a number of birds that had acclimation studies that looked at BMR or INSOM before and after acclimation. And then we used a GLM approach to test whether individual birds that had more flexible basal metabolic rate or summit metabolic rate also had high initial basal metabolic rates. And so here are some data testing that hypothesis from house sparrows. And on the left-hand panel is the basal metabolic rate. I'll note that for when we measured flexibility in basal metabolic rate, that could be either be an increase in the cold acclimation group or a decrease in the warm acclimation group. So both of those are elements of the flexible response. And so what we found with the basal metabolic rate is that those birds that had higher flexibility also paid a cost and had higher basal metabolic rates. This wasn't true, though, for summit metabolic rates, and birds that had high flexibility and summit metabolic rate had a basal metabolic rate that was similar to those that had lower values. And this is actually a feature that showed up for several birds. And so here's the summary of the data from the different studies. And the left-hand panel there shows the flexibility in basal metabolic rate and whether that was a cost. And for 3 out of the 6 birds that were acclimated for at least 3 weeks, we saw a positive relationship between BMR flexibility and basal metabolic rate. For some other species, we didn't, and for the Chinese Huaimei, they actually showed a negative relationship there. So it wasn't uniform, but it was a common element. For the differences in summit metabolic rate, though, we didn't see anything except the junco was close with the positive relationship there. A couple of things I want to point out here. The species with the asterisk there were data that was generated out of Francois Vezina's lab, and it was trying to look at the rate of change in metabolic rates, and so they only acclimated them for 8 days. We didn't see any changes in those birds, and so it might be that it takes longer than 8 days to get the full acclimation response. And then the second thing that I want to show is that you're more likely to get a positive response when you're using colder temperatures for acclimation at the cold end. Okay, so where do we go with this in the future? Well, one, I think we can do better at evaluating the costs and trade-offs. For example, looking at the relationship between metabolic flexibility and investment in the immune system or an oxidative balance. Also, I think most of the acclimation temperature experiments to date have dealt with chronic exposure to a stable cold temperature. So I think introducing temperature fluctuations into that, both over the short term, and Francois' lab has looked at that a little bit to look at the rate of change, but chronic sort of long-term fluctuations in temperature are basically unstudied. And that deals a little bit with this climatic variability hypothesis, which proposes that birds that live in more variable environments should have higher flexibility. And that's been tested in a few birds with variable results, but mostly with relatively minor differences in temperature variation. And then finally, I'd like to see some more work on the potential for muscular non-shivering thermogenesis, which there's increasing evidence showing that it occurs in chicks and growing birds, but less in adult birds. And so I'd like to see some studies addressing the contributions of this non-shivering thermogenesis to metabolic flexibility. And so I think I'll quit there and acknowledge the many collaborators that participated in this work, including postdocs and graduate students, and acknowledge the funding source. And thank you for listening.